As I said, there are two different types or classification of cells, prokaryotes and eukaryotes. So the cells falls under these two major categories. And if you see about prokaryotes, archaebacteria and eubacteria means bacteria is a sole member of prokaryote. But in the case of eukaryotes, starting from a button mushroom to a large whale or animal, which I mean to say, is all of eukaryotic cells. So major cells falls under these categories. And apart from cell, if you look deeper inside, there are called organelles. So organelle is nothing but it's a bounded membrane, so membrane bounded substance which present in the cell, which is essential for the function of the cell, and it independently also involved in its own function, but it's coordinated to form the function of cell. Okay, and here I made a summary of all the organelles, that means major organelles which are contributing to the cell function and how far it's important. And this green arrow means a green tick mark represents it's present in that. If not, this red cross mark symbolizes that it is not present there. Okay, and this is a classification. It's well understood from its own. Fine. Let's go into each and every organelle and a major outer membrane. First of all, just think until what is called as a home. A home is called until you have a boundary. Until you have walls, you can't say you are not independent. That's what evolution happens. As soon proteins or lipids form, unless a cell membrane forms, it is not independent. It should depend on something. So cell membrane or a plasma membrane, a boundary or a cell wall is essential for making a cell independent. So this cell wall is present throughout, means a cell wall or plasma membrane is present in all prokaryotes and eukaryotes. But these animals alone lost this capability of having the cell wall because plasma membrane is well enough for them. The function of the cell wall is to production and structural support. As a plant cells or an eukaryote are exposed to an major environment, they need a physical support or a production to make them hard. So plants, except animals, all other organisms possess that cell wall. And what's the function of plasma membrane? Plasma membrane controls the movement of cells in and out. So unless you have a watchman or a security guard, everyone can come in, right? But what's the use of that? So this plasma membrane, with the support of some proteins present in that membrane's thing, they are doing the job of possessing. So this seems to be essential for all the organisms, starting from bacteria to animals. Then let's go to nucleus. As I said in the initial of my lecture, three things are very much important, and the one among them is the nucleus, means a genetic material. If the genetic material is bound by a membrane, it forms a nucleus. If it is not bound by a membrane, it's not a nucleus. It's just a genetic material. If you look carefully into the meaning, or if you just classify the meaning of eukaryote or a prokaryote, it actually means by the Latin term of true nucleus. Karyote means nucleus. Eu means true, the Latin term. So the true nucleus here, it means having a boundary of nuclear membrane. That nuclear membrane is absent in prokaryote. If it is absent there is no intervention. There is no intervention between nucleus genetic material and the cytoplasm. The cytoplasm lies between cell wall and your genetic material. So hence nuclear membrane is lost here. There is no intervention. It's very obvious. But it's present in all other eukaryotes, the whole eukaryotes. This is the major characteristic feature and a difference between prokaryote and a eukaryote. But if you consider, there are two genetic material, major genetic material, deoxyribonucleic acid and ribonucleic acid. The structural importance of these two things we can see in the molecular biology lecture very briefly. Okay, fine. And DNA is nothing but a hereditary information storing material. So I look like my father look like my grandfather, and definitely my son look like me. So what carries here? The same thing Grieger Mendel observed from the pea plant genetics. He termed that as factors. Later on, they are termed as genes. So those genes are present in the genetic material like DNA. So that encode the hereditary information, which is carried to generation. That hereditary information should mandatorily present in all the species, means all the varieties of start from bacteria to animals. Then I move on to RNA. In eukaryotes and prokaryotes, we are having a single stranded RNA. RNA is a catalytic molecule which carries the genetic information to a protein kind of thing. So proteins are workhorse of our body. As I said, or if you heard of other lectures, you may be aware of what is the function of protein. Start from your color to your function, means my color melanin is due to a protein. My oxygen which I am inspiring, which is carrying oxygen to the entire body cells, hemoglobin is a protein. So proteins are the workhorses of your body. So to support that workhorses, to generate that workhorse, this RNA acts as an intermediate between protein and genetic material called DNA. So this what I have mentioned here. So by a process called transcription, this DNA is converted into RNA, and by a process called translation, this RNA is again is synthesizing protein with the help of machineries like ribosome and other things. Then let us go to ribosome. So ribosome is a protein synthesis machinery. So ribosome is essential for protein synthesis, and it is present in everything. If you observe in my molecular biology lecture, you can find the finest details of how ribosome with RNA or DNA, how ribosome with RNA translating into amino acids. Just imagine 4-letter code ATGC, adenine, thymine, guanine, cytosine is converting into, means in cytosine, this you should observe here, thymine is present in DNA, but in the case of RNA, instead of thymine, it is replaced by uracil. So how this AGCU is converted into 20-letter amino acid code? It's of 64 codons that we can learn later. Then I just go into endoplasmic reticulum. This endoplasmic reticulum is very much essential for lipid synthesis. And endoplasmic reticulum, if you observe in a pictorial representation, it is present around the nucleus. So those endoplasmic reticulum serve an initial modification of protein synthesized from the nucleus. So it's very well organized. As soon the proteins are synthesized in the cytoplasm out of the nucleus, it is modified initially, and the proteins are the polypeptides. And after that, but that all organelles are absent in prokaryote, because there is no proper organization of nucleus, right? So all the absence of organelles are also there, but it's present in the whole eukaryotic sector. Then I move to lysosome. So lysosome is very much, not lysosome, first of all Golgi bodies. After endoplasmic reticulum, Golgi body play a major role in final modification of that proteins, lipids generated from endoplasmic reticulum and ribosome. So after the final modification, it also plays a role in sorting and packaging. So this Golgi body, in a general term, on a civil basis, I will just explain. Golgi body is a postman of your cell, and ribosome is a protein synthesizer, is a factory, is an industrial factory in your cell. And mitochondria, if you go to lysosome, it's called intracellular digestion. So he is the killer, who helps to he is a terminator who helps to terminate all the unwanted thing. Extracellular digestion happens here. Not intracellular digestion happens here. Unwanted cellular materials are digested with the help of lysosome. And mitochondria is generally called the powerhouse of your cell, because it produces the currency called ATP formation. It's like RBI, means Reserve Bank of India, which produces the currency for the entire country. This bank produces the currency called ATP. whatever the energy metabolism happens between cells is ATP serves as a currency, major currency, more than NADPH or anything. So that ATP is synthesized through mitochondria, through lot of metabolisms. That ATP formation is present obviously in all the eukaryotic thing except prokaryote. It's not present in prokaryote. Similarly, chloroplast. Chloroplast is present in green alga and plants, which is not present in any other thing, because green alga is a precursor of the present day plants. Cyanobacteria, which is present in the deep oceans or floating in the oceans, just started the photosynthesis a very 3.5 billion years ago itself. That creates the oxygen to the entire world. That only makes to the evolution of the green plants now, or even the evolution of the eukaryotes which depends on them. So that chloroplast is now present in the green alga of eukaryote and plants of the eukaryote. Then I move on to vacuole. Plant cells are often big due to the presence of vacuole. If you see a plant cell, observe a plant cell under a microscope, you can find a large space, empty space. That empty space is due to fill of vacuole to increase the cell surface. So it's very obvious for plants, and it's in fungal cells also contains that vacuoles, but it's absent in other things like protista, animals, and in prokaryotes. Final thing is flagellum. Flagellum is a unique feature for prokaryote. It's absent in other animals. In some green alga like Chlamydomonas can contain the flagellum, but very few. So flagellum serves as a unique feature for prokaryotes. So this is a summary of all the major organelles in cells. In couple of minutes we can see deeply how each organelle and the finest details of each organelle. So